You're going to learn some amazing stuff. This is absolutely at the forefront, the cutting edge of science right now. It's not your grandfather's genetic engineering in the sense of taking two genes and splicing them together. This is a radically new notion. People are actually composing life. What they're doing is they're writing DNA, and they're writing it the same way that a software developer would write code for a computer. Except in this case, they're writing DNA that's then put into a living thing and can replicate. And some of the breakthroughs are quite amazing. So there's already a drug that's been introduced into the market through synthetic biology, and it has the ability to reach up to 500 million people who are currently suffering from malaria. Hi, and welcome. Ooh,
thank you.
Tricky chair there. So welcome to Chicago, where it's freezing cold. You just joined Autodesk, and I want to hear all about this. So tell me what your new title is.
My title is Distinguished Research Scientist with the Bio-Nano Programmable Matter Group at Autodesk.
That's a mouthful.
It is. It's a conversation starter.
Can you tell us what that means in plain English?
Really, the short story is we're working now to envision what the software design tools might be like for really small and potentially living things.
Autodesk is a company that we tend to think of as a company that's doing 3D technology for architects, designers, and also 3D technology for like computer graphics and special effects for movies.
Exactly.
So how is Autodesk getting into like designing microbes? Is that what you're doing?
Well, really, we make software design tools, and software doesn't care about the scale of matter. Mm-hmm. We need tools for being able to understand, visualize, simulate at really all scales, whether it's a building or whether it's a microbe.
Okay, so in one respect, what you're doing is going to help this citizen science movement because you're building tools that are going to make it much easier for people to compose new life on a computer and visualize what that might be. Is that right?
Yeah, it's all digital design these days.
Wow, cool.
Except now we're working with a different type of computer, really bacteria. They're like information processors that do chemicals, but they're different. They're programmable, but the cool thing is they also make more of themselves. So they're like computers that can reproduce. This is pretty powerful.
That's amazing.
And if you look at a bacterial genome, it has about 5 megabits of information. Really, a small photograph. But this is enough to program a living thing. And really, if you've got a software program, you have to learn the basics. You have to learn how to read it. You have to learn how to write it. Right. We've been sequencing with devices like this. This is a desktop. It's about the size of a laser printer, but it can do a whole human genome. And reading it, we're starting to comprehend more and more of the basic tools of life, this is a simulator for the first bacterial cell. It's actually
what's
really
going
cool.
on? What is this a picture of?
We're actually modeling all the chemical processes that happen in a single bacterium and understanding what's going on below the hood.
So this is a computer model of a cell.
Computer model of a living cell.
Wow, that's incredible.
It's the first time we've ever done this. This was published by Stanford this last summer. And really what we're doing now is we're starting to learn how to write code.
Thanks.
We started writing genetic code back in the 1970s using something called recombinant DNA technologies. It was cutting and splicing genetic code. And really, if you take a look at it, you end up kind of writing ransom note style. You can write anything, but it takes a long time to do. The shift with synthetic biology, and what makes it so powerful, is now we can actually use computer software. We don't need a lab anymore. We don't need these tools, which are really restrictive. We can start using laptops.
So they virtualized the lab.
Yes, essentially now we have word processors for DNA. And the cool thing is we have printers. Companies like DNA 2.0, and there's a bunch of other ones, will print out your DNA, kind of like a Kinko's used to, prints out color copies.
So the analogy to the computers is actually total.
It's really close.
So the idea, in the 80s when personal computers got good, we went from desktop, word composition to desktop publishing, and that was a big change in the print business.
Now we're getting desktop biotech. And so it's
Unbelievable.
push-button DNA, and this is...
So you can compose DNA using a browser.
You can compose DNA using a browser.
You hit a button and send it.
They'll send it off to you.
That's awesome.
And this is opening up biotechnology to a whole new generation of people that are younger and know how to work with computers in the same way that 30 years ago kids started playing with personal computers.
And this is a big part of what you're doing. When I first met you, we were in Silicon Valley, and you were telling me that you've been working with high school kids and even grade school kids to try to get them to to learn about synthetic biology, to maybe give them the confidence to play with it and experiment?
Yeah, I want them to realize this is potentially going to be a field that they will work in for the rest of their life if they start getting interested in
And
it.
how's that going? I mean, do kids respond to this
It's
stuff?
going really well. Really, some students naturally pick this up and they love working with it. And
So they don't see a big barrier. The way I think when I was a kid, biology was for geeks. It was a challenge. You had to go, had to wear a lab coat and you had to
Exactly.
schedule time, and there were beakers involved and so on.
yeah, it was more sciencey back then. Now it's more of an information science.
That's cool. And then do they use these things like when you work with kids? Do they use genome compiler to go like print out their organism or their DNA that they
They
develop?
certainly use these types of software tools and these DNA printers routinely now.
That's kind of cool.
Yeah, it's really sharp.
Take us a little bit into the future. So tell us what's coming next then with this.
Well, really, what's been happening is we're starting to move into modularizing this type of genetic code, making it easier to assemble and to put together faster and cheaper. It's kind of like Lego bricks. It's really extensible. And the groups that's driving this forward is out of MIT. It's called International Genetically Engineered Machines. And it's really it's really the best student program for learning how to do synthetic biology.
Hang on a sec. So it's genetically engineered machines. So they're thinking about life as a kind of like a robot or a machine that you can control.
Yeah, we look at simple bacteria as really being little robots that are programmable.
Do you have examples of that? I mean, have they really built some of these machines?
Absolutely. And this picture shows the 2012 leaders in the field. This is the World Championships. There were over 2,500 students from 200 schools that participated this year. These were the best. The types of projects that I really like showing people, though, this was actually from a few years ago. It was some students at Cambridge University, and they were making super bright glowing bacteria in all different colors. And what they ultimately want to do is to make glowing trees so we don't need street lights anymore. I think
incredible.
That's really a cool idea.
So those are the glowing trees in the background you can see.
Exactly.
That's a cool idea. What else have you got?
Well, this was the one that won the grand prize this year, and it was a group out of the Netherlands, and what they made was a bacteria that could sense when food was spoiling. They said, why are we trusting expiry dates? Why don't we actually look for food that's spoiling and the volatiles they put out? They figured this will save billions of tons of food,
Cool.
annually from going to waste.
So one of the things I'm hearing you say is that when you let lots and lots of people work on these problems, they start finding interesting problems. I mean, the food expiration date probably doesn't strike any of us as a big problem. But you're right, it's a huge waste of food, isn't it?
Exactly. So much of it just gets discarded just because the date, says it's expired. Really, where things are going now is it's actually coming out of academia, and it's this type of biotechnology is coming into the community. This is a lab in Brooklyn called GenSpace, where it's a subscription model for about $100 a month. You're part of the lab, you can take courses, you can use the tools, and get to see some really awesome speakers come by as well.
So that's cool. So that's a little bit different from, in the opening comments, I talked about people who are building labs in their houses, in their dens, or their closets.
There's definitely a few people that have done it.
In a way, we're moving away from that now, where you can go join, it's like joining a hackerspace or a maker community.
It's more fun when you do it with a group anyway.
Sure, and you can get your questions answered, or somebody can show you how.
Exactly, and you don't have to worry about the FBI necessarily beating down your door. So, and the groups that are really getting good at this, well, they're using robots to do not just printing of DNA, but actually booting of the organism and testing. So this is allowing people to really have biotechnology done remotely.
Wow,
just
cool.
Send a design, they'll build and test.
That's a big one. So hang on, that means you could design an organism on your desktop computer, your laptop computer at Starbucks even, and then send it out to get not just printed or not just have the DNA compiled for you, but you could actually have the organism made, manufactured, produced? What verb
tested,
do they use?
and the results come back to you, and you can decide whether you're going to.
So if you didn't get it right the first time, you can do it again.
Do it again.
Is this affordable or is it really expensive?
It's expensive right now, but the prices come down really fast with these technologies.
So like all technology, the price keeps dropping.
Yeah, it drops by about half over some very short period of time.
Wow, cool. And are you seeing like a lot of take up with this?
Yeah, the field's really growing quickly.
Because we keep hearing about it.
And it's bottom up. Like we really started building very simple things when this technology becomes available, because you're actually writing genetic code base by base, letter by letter. And so the types of things people built first were things like viruses that are very, very tiny, just proof of concept. State of the art right now is doing things like bacteria. And only one bacterial genome has been completely synthesized and booted, but there'll be more. And right now, we're moving up into doing more complicated genomic structures. This is baker's yeast. It's just, the foundation of bread and
Beer.
beer. And beer is beer is the most important part, because some people believe that beer is the root of all civilization, and I think it's actually going to be the gateway to a lot of synthetic biology as well. In fact, one of the iGEM teams, one of my favorites.
Here are the German brewers.
I love this. These guys were at iGEM this year. They built 4 synthetic yeast. One of them was a flavor, one of them was a sweetener, one of them was a cancer drug, and my favorite was they put the biosynthetic pathway for caffeine into yeast. So caffeinated beer, and I love that idea.
That's great. So it's beer that brings you up and then brings you down.
Exactly.
Cool.
This isn't old recombinant cut and splice genetic engineering. This is from the code up, every base pair controlled, and we're starting to be able to envision doing things with this, like bringing back a woolly mammoth, or as you said, perhaps even Neanderthals one day.
Well, it wasn't me. That was George Church at Harvard who said that recently.
And he's saying, the technology is starting to allow us to think about doing this type of stuff in the future, not necessarily advocating doing it. The point I like to make is natural selection, the stuff that we were taught in school about Darwin, it's kind of running at, it's kind of reaching the end. Natural selection had 3 rules: eat, don't get eaten, and ultimately have babies, reproduce.
Right.
Now we've got human intention coming into that mix, where we actually have to take some responsibility for the organisms that we choose to create and the ones that we allow to live.
Wow, that's a pretty big point. I mean, that's pretty profound stuff. So you're saying that the Darwin model is coming to an end because human beings now have the ability to direct evolution.
We've been, directing evolution through agriculture, through re-engineering the world for a long time. This is just taking it to a whole new level.
Okay, well, on that note, that brings me to a thought, which is that there's some pretty heavy-duty, I'd say some pretty profound moral implications that I'm not quite sure we're prepared to talk about. Let's invite William Hurlbut up here. So our next guest is part of the faculty at Stanford University. He is a medical doctor. He's also trained in biology. He's got studies in theology as well. And so please join me in welcoming Dr. William Hurlbut to our show. Hey, thank you. Hi. Welcome. So I'm interested to talk about this concept of bioethics, or a bioethicist, I guess, is how we were introduced. And so what is a bioethicist? What does a bioethicist do?
Well, it's an amazing era we're in. We've gone from the mule to the Maserati in the last century, and here we go with this. Raises profound questions about nature and human nature, what's good human purpose. Darwinian evolution may have been largely governed by the principles you mentioned, and mechanism was information, but the product was mind and personal meaning. Yeah. And that's what bioethics is at its core. Now, bioethics has become a discipline within the universities and a dialogue within the society. I personally think bioethics is not so much a profession as it is a conversation, one that requires the whole human family to participate.
Okay, so you're saying that we're moving away from the realm of pure scientists, where maybe there's a conversation happening in academia or in a research lab someplace, and you want to open that up to other people, other stakeholders, people who might have religious faith, or people who might have different sets of values, or maybe frankly people who just aren't as enthusiastic about the future as me or Andrew might be. Is that the notion behind bioethics? Yeah, that's true.
For one thing, in America, public funding of science is a major driver of science,
right.
and it requires assent of the governed population. And there are no small number of people that believe this stuff is apocalyptic, that it's challenging nature, it's eroding fundamental principles in the created order. And I think we have to take that very seriously, and I think there's some genuine concerns in those realms of thought. On the other hand, we have a whole history of retooling nature to higher purposes, and I think there's positive to be done too. So it's a delicate conversation.
But in the past, that happened at a stately pace. So there was time for the regular population to catch up, even if there was some innovation happening, some scientists were pursuing an idea. But today we're in this phase where the phrase I like to use here is smaller, cheaper, faster, better. Yeah. And some of what Andrew just said, just to recap it, Smaller is the ability to work at a small scale. Faster is that you've got the ability, some of the parts are already built. You know, there's infrastructure in place so that you don't have to go build a whole laboratory or a whole fabrication plant. You can get that in on demand over the internet with a company that does that all the time.
Yeah.
Some of these ideas are borrowed, frankly, from the computer business and the internet industry, where we've already perfected those models. And what this says to me is smaller, cheaper, faster, better means the change is going to come much, much faster. We're accelerating. Are you concerned that we might reach escape velocity here, where the conversation is taking so long compared to the advances in the science that the science gets way ahead of society?
Well, I'm not actually as concerned about that as one might think, because the problems get exponentially more difficult as you get toward more complex organisms, and especially the human organism. What I am worried about is the fact that before it was the external world that was the subject of our inquiry and manipulation. Now it's the living realm. All of living nature has become matter and information to be reshuffled and reassigned for projects of the human will. That is a different relationship with nature than simply reordering the combinations of fundamental elements. You're dealing with living organisms now, and that's raises ethical issues, of course, with regard to animals, but especially with regard to what we do with human beings.
Now, one way of dealing with this is that, at least at the highest level of our government, the president puts together a commission, and you were actually part of the Presidential Council on Bioethics. So can you tell me a little bit about that process? I mean, I don't want to get into like Washington in huge detail, but I think it's interesting for people to know that this is what happens in our government. The president finds out about these new pioneering advances and says, gee, I need to know about that from a strategic standpoint.
It was a wonderful experience. I was one of 18 people asked to come together to talk about what we collectively as human beings ought to do with our advancing knowledge and control of living nature. And that is a huge responsibility. Now, I have to say, whatever you may think politically, President Bush really saw that these were significant issues, deep issues, and he admitted he didn't have the background to understand these, and he appointed a very diverse council, diverse politically too, by the way, to think deeply as we could on an earnest level about where our human future should actually go.
Now, what was the outcome of you were working on the stem cell, that was a very intense debate, the stem cell policy.
It was. We didn't actually, they didn't even announce the council until the morning we met, because it was so tense. They didn't want anybody gainsaying who was appointed. Mm-hmm. So it came out in the New York Times, 2 hours before we met. But it was like being in a war zone, actually.
Oh, wow.
The politics of it was very difficult. The animosities generated, and
so they put together a group of very diverse viewpoints.
They did, and we disagreed with one another. There was a lot of contention, and
okay.
I sat there and I listened to the contention, and I thought, well, why don't we solve this? Why don't we try and find a solution that meets the goals of those with moral concerns and meets the scientific medical goals? So I put forward a proposal called ultra-nuclear transfer that would use nuclear transfer technology to create lineages of cells that had the properties of embryonic stem cells without creating embryos. There were other proposals, and one of them that's now in ascendancy is called direct reprogramming, so-called induced pluripotent stem cells. And that's opened up vast realms, and that's the predominant technology now being used. So for the moment, we used our creative powers to jump over an ethical problem, and that's the way we ought to proceed, because there are serious scientific goals, but there are also serious ethical issues.